All right. So what we're going to talk about now is how has life originated on Earth, on our planet Earth. And to understand that, we have to understand also how Earth used to be and how it's changed. Here you have two pictures. This is Earth, how it would have looked once it was formed 4.6 billion years ago. It's a very old planet. And this is how, obviously, you're familiar, how we're used to see it nowadays right here. But in between, life, so Earth has changed a lot in between in all those 4.6 billion years. And these changes ultimately have led to the creation of some situations where life could have evolved. So how did this take place? Well, this is just a timeline. So going through again, Earth has evolved about 4.6 billion years ago. That's when it was formed. It was basically a complete liquid planet. All the rocks were molten. Lots of very, very high temperature situations where life could not evolve. And then it slowly cooled down, start forming a crust around it. First, there was no atmosphere at the beginning, then an atmosphere started appearing and so on. And life started appearing around 0.6, 0.8 billion years after the formation of Earth, so around 3.84 billion years ago. And the first evidence are around 3.6, 3.8 years ago in the form of deposit from the first known bacteria, which are cyanobacteria. After that, basically, all organisms would have been prokaryotes, so with a single membrane, no internal organelles, simple chromosome, and so on. But then, about 2 billion years ago, we started having the first eukaryotes and the first multicellular organisms appearing. And these are, or multicellular eukaryotic organism appearing. These will start having organelles inside, more complex DNA, multiple chromosomes, linears, and so on. And then animals started appearing about 0.5 billion years ago. And we have been here on Earth just basically from a few hours. If you think about the big scale, so how long the Earth has existed, and your book shows it very well. So if you take the overall scale here, you put it like, assuming that this is a full year, we would have existed just for a very few seconds, for a very few minutes here at the very end of the Earth life. So it seems like we've been around for long, but Homo sapiens, or humans in general, have been around for a very, very small fraction of all of this. So first of all, why has life started on Earth? Why nowhere else? And ultimately, it's because Earth is what we call a habitable zone because of some characteristic which makes it, that basically makes it a stable environment. Ultimately, all the properties that led to the formation of living organism they lead to stability. What are they? First thing, Earth is located at a proper distance to the sun, very good distance, which basically allows for the water present there to stay fairly stably in a liquid form. It freezes sometimes, it evaporates sometimes, but most of the water on Earth is always on the liquid phase. And this guarantees stability. Why is stability important? Just to give you an idea, imagine you're gambling, all right? And gambling brings some money there. And every time you gamble, oh, someone changes the rules. Could you really play? No. Same thing for evolution. Evolution needs stability. What evolution is doing is randomly, right, testing different variables. But obviously, to test them, you need the rest. Since these are variable, you need the rest to be stable, right? So you can't test, or you have something that presents is extremely stable in high oxygen, boom, then there is no more oxygen on the atmosphere. And then the following day, the oxygen comes back, or there's more nitrogen. These would not allow for the testing of different variables and therefore evolution to happen. So the liquid form of water basically created a very stable environment where life could have started. Second thing, Earth has a strong gravitational pull. Why is this important? It's important because this works not only on whatever it's on the top of the crust here, but also on the gases around the atmosphere. So the pull actually keeps these gases close to the Earth, creating that mixture of gases that we refer to as the atmosphere. Why is this important? Because this atmosphere now has become, with time, rich in oxygen, ozone, which ultimately has preserved the Earth from all the energy that is in the universe to freely hit the crust. So, the UV rays from the sun, for example, they're filtered by ozone, right? And so by doing that, by having something in this atmosphere, first the formation of the atmosphere and something there that blocks these rays, now you start creating a space on the Earth crust which is stable, where there is no random energy there that would destroy everything. Okay, so the two big things: water could stay liquid at the temperature of Earth due to the location of Earth compared to the sun, and the gravitational pull led to the formation of an atmosphere. So that's the reason why life is present here. Now, how's life has evolved? To understand that, first of all, I want to stress on one thing. What we're going to get into now is just a series of theories, hypotheses. Okay, there are some supporting evidence, but they have never been proven. In science, nothing gets proven. But these are theories that are widely accepted as possible by scientists. So to understand what we'll talk about next, we have to understand basically how would life have evolved. So life would have had to follow a series of steps that ultimately follow the hierarchy of life and the properties of life. So first you would have had this random formation of the biomolecules, which would have not been living organism, but then these biomolecules somehow would start joining together and forming ultimately the unit of life, which is the cell, the first cell. And then from these cells, the cells would have somehow found a way to store energy, and you would have started having some genetic material appearing, and therefore you would have had these first cells would have started being able to replicate, evolve, have some metabolism, and therefore slowly spreading throughout the earth. So let's start with the formation of biomolecules. When we talk about biomolecules, just to be clear, what we're talking about is nucleic acid, so DNA and RNA, proteins, lipids, and carbohydrates. We'll talk about the structure of these biomolecules in the next few lectures. But how did they form? How would they have formed at the beginning of the Earth history? Well, there are different hypotheses that explain how it could have happened. There are several of them, but the main one, the three main ones, are the following. What is referred to as the reducing atmosphere. The second one is referred to as the deep sea vent theory. And the third one is the extraterrestrial origin. Let's start with the reducing atmosphere one. What the reducing atmosphere theory state is, actually, to understand it, first of all, we have to think about how was the Earth at the very beginning. So 4.6 billion years ago, it was all molten. Nothing could basically stay there stably. But then it started cooling down, forming some crust. There would still be big eruptions there, which will release a lot of gases in the early atmosphere. So the early atmosphere was actually very different 4 billion years ago than it is now. Now we have oxygen. In those days, there will be no oxygen. And the molecules present there released from this eruption here will be hydrogen, ammonia, methane, water vapors there. And most of these molecules here would actually be highly reactive. That's what we call the reducing molecules. In chemistry, reducing molecules are molecules that easily can move electrons around. They have lots of electrons, they can move them around, and by moving them around, they can actually form bonds, meaning that they can interact with each other. Okay, so these molecules here will be very prone to interact with each other. However, they will still need some energy. And how would they get this energy is by the fact that, as I said before, at the beginning there will be no oxygen, no ozone, and therefore Earth will be freely hit by, imagine here, by the energy coming from the sun in the form of UV rays, and also lots of lightning and so on. So lots of energy in the atmosphere as well, which then could help this molecule to react with each other. And that's what this scientist here sorry, I'm going the opposite way, Oparin and Haldane here in the early 20s hypothesized is that these chemicals here, highly reactive with each other, reducing compounds, could have interacted with each other, and because of the energy present there, coming from the outside, they could have reacted and formed the first basic molecules, which then later on would have formed the DNA, RNA, and so on. Well, there is an experiment that was done later on by Stanley Miller in the middle of the 1900s, in the year where actually the DNA structure was discovered, which basically what it did to test that Oparin and Haldane's hypothesis, he basically recreated the reducing atmosphere inside a glass enclosure here. Not only did that, so he put all the reducing compounds that would have been found in the early atmosphere, but also he provided energy similar to the one you would have had. In this case, what he was doing is providing the energy through electrodes, simulating lightnings and so on. And then, after doing that, he let this small atmosphere go on for a few days, and then he isolated the compounds through precipitation and so on, evaporation and precipitation here, the compounds that eventually would have formed. And what he actually found, about 1 week later, he found that there were many compounds such as urea, simple amino acid like glycine, lactic acid, formic acid, and acetic acid, compounds that are all found and they're very common in living organisms. They could actually be found here in his experiment. So supporting the evidence that the early atmosphere, reducing atmosphere, thanks to the energy that was coming from the outer space, could have actually led to the formation of the very first basic molecules, which are now the base of living organism. Not only that, but later experiments were done in a very similar way, but with the addition of an extra molecule, which is hydrogen cyanide. This molecule is extremely abundant in the universe, so it would have been abundant in the early atmosphere as well. And what they saw is that by adding this simple additional molecule here, now they started seeing that also, and formaldehyde as well, sorry, two molecules there, again, both of them present at that time. Now they start having more complex compound forms like purines and pyrimidines. I don't know if you're familiar with that, but we'll talk about that in the next few lectures. If you look at the structure of nucleic acids, they're formed by molecules called nucleotides, which, as one of the three components, they have purines and pyrimidines. We refer to them as the nitrogenous bases, or nitrogen-containing bases, or nucleobases. So these could have been randomly formed at the beginning, thanks to the reducing atmosphere plus these common compounds present there. What they also found with these later experiments is that also more complex amino acids and fatty acids could be formed, plus simple carbohydrates like glyceraldehyde, ribose, glucose, and fructose. So all of these molecules, they are critical components in living organisms to form the biomolecules. Now, that's the first hypothesis. That's the reducing atmosphere hypothesis. The second hypothesis is the one that refers to the deep sea vents. And what it says is basically that life could have started in a similar way with some slight differences, not in the atmosphere, not on the crust of the earth, but underneath in the sea, in the water, where you have these basically material escaping the core of Earth through these cracks, which are referred to as deep sea vents. The vents will refer to this black material that comes out. Basically, it's due to the very high temperature, the water very hot coming out of here, coming out, meeting cold water. You have hot hydrogen sulfide gases here coming out, and then you have precipitation that form these black clouds there. Now, this high temperature here and the escape of material highly reducing like methane, ammonia, hydrogen sulfide, which I mentioned here, could have again still stimulate the interaction of these elements, forming again very basic molecules like amino acids and so on. Not only that, but what has been shown recently is that in deep sea vents, you start having also an abundance of hydrogen being formed here, which could have been used by the first cells also to recreate that proton gradients that we will see later on when we talk about cellular respiration, so energy formation, could have been used to form energy, to retrieve energy and use it for metabolism. Finally, the third hypothesis is the one that states that in reality, and all of these hypotheses, keep in mind, could have happened at the same time. So it's not that one hypothesis excludes the other one. The third hypothesis, what it states is that in reality, some of the molecules could have even come directly from somewhere else in the universe. It's possible that basic molecules, organic molecules, could have been formed, such amino acid, basic amino acid, and so on, purine, pyrimidine, somewhere else, and arrive on Earth through meteorites. And to support that, there are evidence that, and by studying some types of meteorites that are right here, called carbonaceous chondrite meteorites, about 5%, I believe, of the meteorites reaching Earth are this type. And they've been studied chemically, and they have been shown to actually contain these simple organic molecules, which are, again, key components of living organisms now. So it's possible that some of the early basic molecules could have actually arrived from outside the Earth through meteorites or other ways. Now, what we talked about until now, so these three hypotheses, the deep sea vents, the reducing atmosphere, and the extraterrestrial origin, can explain how the first simple molecules would have been formed. But these molecules per se do not represent life. So how would we have gone from these simple molecules to more complex molecules like DNA, proteins, the biomolecules? In order to have that, then you would have these simple purines, amino acids, first carbohydrate would have had to interact with each other. And in order to interact with each other, oftentimes you need some substrate that helps them get close together. Right? And again, nobody really knows how that could have happened, but there are molecules that were present in those days that could have helped. And one of them is definitely clay. Clay was very abundant in the early atmosphere, still very abundant nowadays on Earth. And these could have basically prepared a substrate for this molecule, would have precipitated there, by precipitating it there, would have found each other, close to each other. And again, with the external energy, could have started interacting with each other, forming the first nucleotides, the first nucleic acids, first complex carbohydrates, fatty acids, and so on. Now, again, we have not got to the point that we're talking about life. We're talking about biomolecules. How did these biomolecules then give origin to a life? In order to do that, they would have had to start interacting with each other, forming, first of all, the membrane that then define the cell, right? So cell life starts from the cell. So some of these molecules would have had to interact, forming membranes. And then there should have been one of these molecules taking the role of genetic information, not only that, but also as a molecule that could be able to perform some metabolism. And in order to do that, the early random organisms should have been able somehow to retrieve some energy. So how did we get to that? Well, when it comes to the membrane, it's well possible. Now, if you look at the structure of the membranes, and we'll talk about that in the next weeks, is membranes are made by different things, but particularly phospholipids and proteins. And the beauty of phospholipids is that they actually form membrane-like structures like this, so lipid spheres or micelles and so on, by just being together in an aqueous environment. They are partly hydrophobic on their tails, and therefore to do that, they form these complexes on their own without requirement of energy. They do that to escape water by keeping the water outside and in the inside, but creating a hydrophobic environment here. They do that on their own. So the formation of the first membranes could have been simply an immediate event after the formation of the first phospholipids. They could have started forming membrane on their own. And by doing that, they could have started trapping in some other organic molecules like the first proteins, first nucleic acid, and so on. This doesn't mean that they will form a cell. Okay, so they would have formed what we call probionts. So they're basically organic molecules surrounded by a membrane here, but these would not form a cell or a living organism, because in order to be a living organism, you should have some energy production here, some metabolism happening, some genetic material that would allow for this sphere then to produce more spheres. And so these are just probionts, so early form of pseudo life, but they are not life. How did we get from probiont to life? So clay also, forgot to mention here that also clay could have helped with this formation of the lipid vesicles, despite the fact that, again, the phospholipid tend to form liposomes on their own. So these lipid vesicles on their own. Now, how did we go from probiont to actual living organism? To understand that, we have to understand. So this is how life is now. So now we know that every living organism has some DNA, which then contains all the information to form proteins. And this information is first transferred into another nucleic acid called RNA, or ribonucleic acid, which then has the information that in the ribosome gets translated into proteins. And then these proteins, basically they do almost everything inside the cell in terms of metabolism, okay, doing the reaction, the enzymatic activity. Well, this is very complex. Okay, so you have this genetic molecule giving origin to another molecule, then gives. So there is a lot of steps in between. You see when we talk about transcription and we talk about translation. All of this, by the way, is what we refer to as the sense dogma. So that's how we understand life. The flow of life is in all living organisms. What's possible is that in reality, earlier on, the picture was much simpler. What you would have had that basically RNA would have been the first nucleic acid to be formed. There was no DNA at the beginning. And these molecules could have worked. It's very similar to DNA. It's less stable though, being single strand instead of double strand like the DNA. It's less stable, but it could have had, it's made by 4 nucleotides. It could have stored genetic information, similar to how DNA does nowadays. Not only that, but it could have also performed the first early enzymatic reaction, so the very early metabolism could have been formed at that. This theory is what referred to as the theory of the RNA world. So the first living organism would have their vesicles, and inside, right, mostly RNA with the function of carrying both the genetic information and the first reactions. And then these, so one of the reasons why there are evidence that can support that. First of all, RNA again acts already as an enzyme, still acts as an enzyme in some situation. You see when we talk about transcription, so how the information is transferred from DNA to RNA, how this information is copied into RNA is referred to as transcription. There are some processes here in between, and some of them are carried over by RNA itself, where it can cut itself and remove some sequences. So RNA can have enzymatic activity, but now with time, this enzymatic activity would have been passed on to proteins. And then also being formed by 4 nucleotides plus having different shapes that it can assume, it could have contained a lot of information on its own. Then later on, this information would have been transferred to DNA in terms of genetic information. And the advantage of mDNA as a double molecule, first of all, DNA is very similar to RNA. The big difference is in the sugar that you have, there's just an extra oxygen overall. And also one of the 4 nucleotides is different. But altogether, this allows for DNA to become double-stranded and much more stable as a molecule, which gives an advantage to a living organism having DNA as a genetic molecule. So DNA could have become taken over, if you want, the genetic part of what the RNA was doing, whereas the enzymatic part could have been taken over by proteins slowly. How and why? Mostly why? Because if you think about how proteins are made by 20 natural amino acids. You can imagine the combination you can have 20 amino acids. It's very vast. Whereas the nucleotides found in RNA are only 4. So the combination of 4 of them is It's much more. Think about if you're printing scramble and you have 20 letters versus 4 letters, you can create many more words, right? And therefore many more variety, many more different enzymes, and therefore you can carry over many more different reactions if you use the 20 amino acid or the 20 letters in your case. Whereas if you have only 4 letters, good luck, you can create that many words, that many enzymes, right? And so with time, proteins would have taken over. Again, I'm stressing we're talking about theories, all of this. So slowly, basically, we would have got to this central dogma that was dogma where the genetic information is now taken over by DNA. RNA becomes just a transitional, basically, molecule, or mostly a transitional molecule, where the information that's helpful to transfer the information from DNA to the proteins. Next step. So that's for the genetic information. So early organism would have had also the need to be able to store the first energy. And in order to do that they would have needed a molecule that could store a large amount of energy. The good news is that one of the first of the nucleotides, so the first simple molecules formed, ATP, is actually one of the molecules that can store a lot of energy in these phosphate bonds represented here. So one of the first few molecules could have already been adapted as an energy storing molecules, which then later on could have been used to retrieve or to store the energy retrieved from the sun or other components there. And so this would have led ultimately to the first living organism, so the client slowly would have acquired this capacity of storing genetic information, storing energy, performing the first reactions, and ultimately being able to duplicate. Now, the first evidence that we have of living organism is what we refer to as stromatolites. These are these towers that you see here. They're nothing else than precipitate, which is left behind by bacteria of the type of cyanobacteria. These are the earliest bacteria known. These are photosynthetic bacteria, which are also thought to have become the chloroplast in plants nowadays. These are the first evidence that we have of life. And then from here, obviously the first organism would have been simpler than cyanobacteria. This is only what we have as a first evidence, but we can hypothesize that there would have been an earlier precursor as well, which is referred to as LUCA. Sounds like Italian, but LUCA stands for last universal common ancestor. So the last shared ancestor that we, bacteria, all these plants have in common. And this would have given origin to all life, which can be grouped into 3 domains: archaea and bacteria, which used to be classified altogether as prokaryotes, and we still refer to them as prokaryotes, and then eukaryotes as well is the 3rd domain there. So, but all of them, as this picture can show you, would have arisen from a common ancestor here, very first living organism on Earth. Cyanobacteria would have been already at this point. That's where the first evidence we have of life. But they could have been one of the earliest bacteria known on Earth. Now, about 2 billion years ago, then we had this next step in evolution where you had that different type of living organism referred to as eukaryotes would have appeared, and multicellular as well organism would have appeared. It's about 2 billion years ago. And the classical differences between eukaryotes and prokaryotes is that now you have that the DNA is safely stored inside a dedicated organelle, which is the nucleus. So there is a membrane there that protects that. And also you have now organelles that are present in there. There are other differences, like DNA is much bigger, so it can contain more information at this point, divided into linear chromosomes and so on. There are many differences, but these are the key ones. DNA is protected better than in prokaryotes and has a dedicated machine inside there to duplicate information and translate it into RNA. And then you have other organelles which have different functions. So the functions now in eukaryotes start being into dedicated organelles. And these organelles that we're referring to are mitochondria, chloroplast, the ER and the plasma reticulum, the Golgi, the lysosomes. There are many other organelles here, which ultimately would have given an advantage to eukaryotes, because now having different function done specifically in different areas, such as in the mitochondria, now you can just produce energy here instead of on the whole cell. You can have much more efficient productions there. It's like, imagine if you have a company where you have everybody doing everything versus a company where you have dedicated departments that focus only on one specific aspect. Obviously, the company has dedicated departments, as long as they're well organized, will be much more efficient to perform the task compared to the one that has everybody doing everything. Okay, so specialized people can focus on one thing, do it at their best, and then share it with everybody else. And that's the advantage of eukaryotic cells. So how did this organelle ultimately come into existence? Again, we're going back to the realm of theory here, but the main accepted theory, which is very well supported, is the one that is referred to as the endosymbiotic theory, which basically what it says that living organism could have arisen through the engulfment of other organism by a different organism. So imagine this being the original prokaryotic cells. This cell could have engulfed other smaller organisms, right? And once that happened, these organisms, once they found themselves inside, they could have provided an advantage to the original prokaryotic cell, and vice versa. The original prokaryotic cells could have presented an advantage to these engulfed bacteria. By doing that, you would have had a symbiotic relationship. A symbiotic relationship is any relationship where there is mutual benefit, right? And by doing that, basically you would now start having this living organism living inside another living organism almost independently, but still sharing the information. Now, this is fairly evident in terms of experiments and different evidence when we talk about mitochondria and chloroplast. Mitochondria could have arisen from aerobic bacteria, and chloroplasts are almost sure to be arisen from photosynthetic cyanobacteria, those early bacteria. What are the evidence supporting that? Well, there are many evidence that both chloroplast and mitochondria provide to support the endosymbiotic theory. First of all, they both have a double membrane. That would make sense. So originally you would have had one membrane here for the original prokaryotic cell and one membrane from the whatever bacteria would have been engulfed. And then obviously through the process of engulfment, now you start having two membranes, one coming from the original prokaryotic cell and one coming from the bacteria that's been engulfed. And this is exactly what you see here. So the external membrane of mitochondria and chloroplast, and an inner membrane of mitochondria and chloroplast. Second evidence is that both mitochondria and chloroplast replicate independently. They replicate as independent organisms inside the cell. So, for example, if you start with a cell with a few mitochondria, a few days later, you will see that these mitochondria are being replicated. Even if the cell they are in has never replicated, they would have replicated on their own. And why can they do that? Because they have also their own genetic information. They have their own circular chromosome inside, both mitochondria and chloroplast. These circular DNA, circular like in prokaryotes, even though they're inside eukaryote cells, actually contains active genes, 39 in mitochondria and about 100 in chloroplast. Now, not only that, but if you sequence the DNA in chloroplast, you will see that it's extremely similar to the same DNA that is found in these early bacteria, cyanobacteria. We're talking about 99% similarities. So these really support the fact that these could have been bacteria, independent bacteria that have been engulfed by other prokaryotic cells before. Finally, the last evidence is if you look at the ribosome that there are inside mitochondria and chloroplast, and keep in mind these genes are actually active to get translated into protein inside the mitochondria and the chloroplast. And this is done in ribosome that they are of the same type that you find in bacteria or prokaryotes, 70S. In eukaryotes, they are referred to as ADS. They are larger. Whereas in prokaryotes, there are 7S. And the one you find inside mitochondria and chloroplast are exactly like the one you find in prokaryotes, so 70S. So again, striking supporting evidence here for the endosymbiotic theory there. Now, things that could have happened later on, it could have been some transfer of material from the genome of the engulfed bacteria that would have moved into the nuclear DNA of the engulfing cell. And these would have created also a dependency in terms of production of proteins that now can be formed also in the nuclear genome and go back basically into the organelle chloroplast of mitochondria. We have this happening all the time, these passage proteins from the nucleus into the chloroplast or the mitochondria. So again, what was the advantage of all of this, of having eukaryotes, and why did they spread so well later on Earth? Is because they're definitely much more energy efficient. Having mitochondria specialized units for production of energy gave them a great advantage, made them more able to be more efficient to retrieve the energy from other molecules, particularly glucose. And together with that, also having different organelles specialized or to do different functions, like for protein synthesis, lipid synthesis, and so on, digestion of engulfed material, made them also much more efficient compared to prokaryotes. And this also would have led later on for improved specialization and the evolution of multicellularity there. So in summary, with both this recording and the previous one, what have we discussed here and what I want you to take home? We talked about what defines living organism. What are those properties? You remember they all follow the same hierarchy. They all have genetic material, they need energy and so on. Then we talked about how life has started. So how did we go from basically nothing on Earth or no living organism to the formation of the first living organism, or the last universal common ancestor, LUCA or LUCA. And so we talked about the three theories explaining the formation of the first molecules, the deep sea vent, the reducing atmosphere, and the extraterrestrial origin of these molecules. And then we talked about how we went from there to the acquisition of basil-like properties. So the capacity of storing energy through ATP, the first genetic material being RNA, not only genetic, but also enzymatic molecule in the RNA world theory, and so on. And then we finally talked about how did we go from prokaryotes to eukaryotes about 2 billion years ago, 2.5 billion years ago, and through the process of endosymbiosis, and what were the advantages of eukaryotes.